The webinar is now live. And the participants are flooding in, which is lovely to see. Thank you everyone for joining. We'll be getting started in a couple of minutes. We'll just allow a minute or two for everyone to register and get into the webinar.
Brett, master of timekeeping, are you giving us the go when it's time to start?
Oh, that's a very good point. Yes, we should probably kick off as it is 3 o'clock, and I will try and keep us as close as possible to time. So away we go.
Okay, cool. Hi, hello, and welcome everyone to this second virtual podium, Asia Pacific, for and by the Metabolomics and Lipidomics community. I'm Anne Wendt, I'm from the Singapore Lipidomics Incubator, and I'll be hosting today's session together with Peter Michael from the Baker IDI. And I'd also like to give a shout out to the other members of the organizing committee, Belle, Brad, Fahana, and Tusi, and also to Kathy and Sarah for creating this opportunity. The format of today's session is that we have, we start with a keynote session by nature of 15 minutes followed by a short discussion. And then we have 6 lightning talks today of 5 minutes each, followed by a joint Q&A for all the lightning talks. And then we have a second keynote presentation. And after that, a joint Q&A session for all speakers together. So here you see that we are quite happy that we have lightning talk speakers from all over Asia Pacific, also covering a wide variety of topics and biological questions. And with that, I hand over to my co-host Peter to introduce our first keynote.
Thanks, Anna. Once again, it's my pleasure to introduce the first keynote speaker for today, and that is Dr. Nirja Kanani. So Nirja is a senior principal investigator at Singapore Institute for Clinical Sciences at A*STAR. And her research focuses on using integrated omics approaches to identify biomarkers and interventions linked with metabolic and mental health adversities. And she's particularly focused on both pediatric and adult populations. And so Nirja's talk today has the title of Human Circulatory Lipidome, Insights from Iomics Studies in Pediatric and Adult Populations. So over to you, Nirja.
Thanks, Peter, for the introduction, and also many thanks to the organizing committee for putting together this wonderful virtual podium. It's a big challenge to bring scientific communities together during the COVID period. So really well appreciate this. Looking forward to more talks as they come in the next sessions. So I'm going to share my screen and let me know. Are you able to see the screen?
Yep.
Okay, and we're going to go in the PowerPoint mode. Hey, are you good with?
Yep, looks great.
Okay, all right. So as Peter mentioned, our lab is located in Singapore. And those of you who visited or know about it, it's a small island nation of 6 million population. And its ethnic component is primarily Asian. And that's the reason Singapore is also a hub for Asian-centric translational studies. And the main areas of healthcare are pretty much common to what people are witnessing as global issues in the developed nations, that is metabolic and mental well-being. Singaporean kids at school-going age, 12% of them become obese, and the adults, 1 in 11 develop type 2 diabetes, and the numbers are expected to go up to 1 million by 2050, which is pretty big a number for a small island like that. And then mental health is again a bigger challenge, with 1 in 16 developing mental health adversities. And now with the pandemic going on, I think both metabolic and mental health challenges are at its forefront of healthcare, and there would be dynamic changes happening that we need to pay attention to. And so our lab's work is pretty much catered to all these health adversities that we're witnessing at the moment to increase. And what we do is look deep into the Asian phenotypes. Now, even though we say it's a common health challenge across the globe, but we do know that populations are different. The phenotypes can arise in very different ways. There are different pathophysiologies to it. So what might be obese in one population may not be in the other. For example, if you take the Indians among the Asians, they might not show a high BMI increase, but they might have a higher level of ectopic fat accumulation that leads to metabolic adversity. So they might accumulate more fat in abdomen as well as in the muscles. So clearly there are different phenotypes that feed into common ailments, but we have less understanding because of the knowledge gap that exists in different variants that people have been mapping, or the scientists have been mapping, whether it's the genome, it's the lipidome. And so what we're trying to do as a group is to build more precision in these molecular subtypes for the Asian communities, so that we are moving forward with precision in our interventions and diagnosis. And another important thing to know is it's just not the population differences, but then it's what is the right time for intervening. And a lot of studies are done in the adults, but there is little work on the younger subjects, that's in the pediatric population. And so our focus is also on them. So we move both of them together, compare them, and see when is the right time to intervene, and can we break the cycle of transmission to the future generation. So we do a holistic approach of studying women and to be fathers during preconception, pregnancy, and when the child is born, longitudinally look into them. They're in our cohorts, they are in adolescent stage, but then we also see what we're finding from there, compare it with the population studies and government-based national health platforms to see how does our finding look there. So it's a holistic approach, and along those lines, we have engaged with multiple cohorts and national platforms, as I said, and we published quite a bit along these lines in past 7 years of work that's been done in Singapore. But for today, what I'll do is give you a peek into into some of the biomarkers that we are looking through different omics platforms and how we find that human variome studying at population level and in different age groups can be helpful in bringing precision. So coming to human variation, there are different ways of mapping them. And people have their favorite omics platforms that they use, which can include looking into fixed genetic variants, but we know like genetics doesn't explain everything. And so there are other variants that we need to map. And for example, there can be changes in the DNA methylome, that is the epigenetic changes, or the transcriptome, which is like real-time capture of like if the transcription is changing, then accordingly the proteins are changing. And then the lipidome, which is one of the central focus of this podium. How does that vary? And, you know, knowing that each of them are not sufficient and they tell you some part of the story, it's important to integrate this information, which is quite a daunting task, but the interactome can be more informative if you know, we have the information and if it's done right. So along these lines, I'll give you some observations that we have had by studying in our community. So Singaporeans, as I said, like are primarily Chinese, Malay, and Indian. And so we did this mega effort of mapping the genome of 5,000 Singaporeans. And this was the very initial study. It's a consortium called as SG10K. And in that, what we found by mapping these 5,000 genomes is that we do have a very discrete genetic makeup. So this is a typical PCA plot of the SNP variants, where you would find that we have the Malay, the Chinese, and the Indian, that they tend to segregate quite nicely by their variants. So clearly the genetic pool shows diversity. And the key findings were like, you know, if you compare it with what has been available through the human genome maps, and by doing the Singaporean genome, we did find that there were 52 million novel variants that were missed in the previous analysis that have been attempted in Asian communities too. For example, we do have some Chinese and Indian SNP variants that are available in the genome map, but not necessarily it is exhaustive. And then besides the new variants, we also found certain loci which are under ancestral selection. So there are 20 loci, and then 14 of them, that the new ones that we found, they were linked with these complex traits involved in the non-communicable diseases. And more to the point, I mean, the more deeper and the dense the genotyping is in the different communities, it helps also to impute these variants well in the Asian population. So overall, I think what we've been doing is doing a dense genotyping of our population. And then what we've also done is not only just look into genetics, as I said, we want to look into the other variants. So that's DNA methylation as well as transcriptomics. So what we do is we profile them. The data that I'm going to show you is being done in 900 women from a birth cohort study, which is called GUSTO. And we have a fair representation from all three ethnic communities. And we've done extracted the blood cells, and we have looked into these different variants. From the variants that we find, we've split them into two parts. One is those that can be environment responsive, that is, they are not genotype dependent, and those that are genotype dependent variations. So accordingly, we call them as two forms that you would see. So SNP alone influence a phenotype, but then a nearby SNP to a DNA methylation site can influence the methylation at that position. So they are called as methQTLs. And likewise, a transcript can be under an influence of a genotype. They're called as eQTLs. And so we mapped these QTLs, the quantitative trait loci. So these are SNP and there another layer that adds to the missed knowledge gap that add to the knowledge gap of what we know from different communities. And then we also mark the non-QTLs, which is could be environment induced and not genotype dependent. So if you look into the genetic mapping of these women, we find that again, as I showed you, that Chinese, Malay, and Indian tend to segregate well by the genotypes. If you look into the DNA methylation, we find similar observation, although the level, if you look into the PCs, it's not one of the topmost component that segregates it, but definitely we do find Indians tend to be quite different in their methylation profile compared to Chinese and Malay. And the same goes for transcriptomics. The resolution isn't that strong here of these groups, but there are certain transcripts in the blood that are variant between the ethnic groups. Okay, so the question comes like, so what's the relevance of studying this? Where are these variants, in which genes are these variants located? So when we do the profiling, we map them to the genome, and now we look into the genes where these DNA methylation, transcription, as well as the SNP variants lie, and we try to find the hotspot genes which show more ethnic diversity. And so we identified 671 genes that have these this variants very dominant, coming from all three measures that we did. And when we look into the gene pathways on which these genes fall, the topmost pathway that comes out is lipid metabolism. And so that led us to go and profile the lipidome of these women, which thanks to Marcus and Peter's team, which we did as a combined project, we looked into it, and we do find that there is downstream difference by ethnicity in these women for their lipidomes, their circulatory lipidomes, which is evident here, so the orange being the Indian, but we do find that the Chinese and Malay tend to have way more similarity. Just to give you an idea of, well, we see these differences in the blood, but does the variant that we found actually explains the lipid variation in the blood? Here's an example which we take of measures of plasma arachidonic acid and the SNP variant that is FATS1. So FATS1 is a known gene in omega fatty acid metabolism and plays a role in arachidonic acid levels too in the blood. And so we found that FATS1 SNP was different. So let's have a look here. So if we compare the arachidonic acid levels in Chinese, Malay, and Indian, we do find the Indians tend to have in the blood higher levels of arachidonic acid. If you look into the FATS1 SNP, if you look into the FATS1 SNP, we find that arachidonic acid levels tend to be higher in subjects that have a GG phenotype, genotype compared to the CC. And when we compare the three ethnic groups, we do find that Indians tend to have a higher GG genotype representation in the population, so that gives us an idea that population diversity in the genotype is linked to the lipid level. And so that raises a major question, is that if we see in a population a difference in the lipid profiles, is it a risk for them? Because arachidonic acid is known to be a pro-inflammatory marker and is linked with many mental health related cognitive diseases as well as metabolism. And so is it a risk or is it an adaptation? Because this thing has been there over generations and it's been as an ancestral legacy to this particular thing. And so the question is, have over time Indians adapted to it? So what we've done is we've taken this a little bit further and we have gone and looked into the pediatric versus adult comparisons also of these lipid-based QTLs. And so here what we've done is comparing adult versus child at different stages, whether it's birth or at 6 years of age, and we mapped it at different time points. And so we mapped the lipidome of these individuals at different stages in the life course. We find that at birth, the lipid profiles are very different than what is it as a child grows up at 6 years, but it starts becoming similar to that of the mother or an adult. And for a woman during pregnancy and non-pregnant state, also the lipid profiles are very different. So we went ahead and we mapped SNP variants that could be linked with these changed lipid profiles, and lo and behold, we do find a difference here that if you compare different stages, there are some common lipid QTLs that pop up at all stages in physiological states, but then there are new ones that we see in cases of the child at birth. So this is just showing that if you go by, so chromosome 11 has the maximum commonality of the variants at different stages, but there are certain chromosomes that show birth-specific lipid QTLs. And if you want to see which ones are the common ones, these are again those belonging to omega fatty acid metabolism, but they are distinct lipid profiles and QTLs that belong to each stage. And this is a summary of like the 24 lipid classes and the different number of variants that we picked in each at different stage. Clearly the adult and the pediatric at 6 years look pretty much similar, but I think what we need to bear attention here is that some of the lipid classes, you will be able to see these QTLs to show their relevance only in the physiological state or an age group. So coming to the end of the talk, I would say the take homes here is that we do require a deep learning of human variation if we need to build precision. What time of intervention do we really need to get into? It will depend on if these variations in the lipid profile are mapped in different physiological states and the stages of life course. But outstanding questions that such studies raise is that are ethnic differences in lipid profiles, they are risk factors or adaptation. And so will interventions work if that's their normal profile and elevated arachidonic acids in Indians? Will that be of any use to change it? And then also the current clinical reference ranges that we've been using as generic guidelines, are they population specific? Do we need to revise them? And I think Marcus at some point had alluded in his talk last week that we really need to get into individual populations and do pull out these ranges which might be specific to them. So I'm going to end this talk by thanking team. We want to know more about our lab, use the QR code up there. But this work today, which I presented, was mostly done by Ben Hong, Chen Li, and Sartaj. Sartaj is a postdoc who's working between me in our lab and Marcus's lab. I also very much appreciate Marcus and Peter's role in help us work on this lipidomics in different age groups, this mega study that we did, and our cohorts, as well as the population study panels. So with that, I'll be happy to take any questions.
Thanks, Nirja. That was fantastic. It's great to see the analysis of all that data progressing, and obviously there's an enormous amount of information in there that you'll be able to tease out. So do we have any questions for Nirja? And perhaps while they're coming through, I could just start off. The methylation analysis and the patterns you got on the PCA showed the Indian ethnicity standing out quite distinctly from the Malay and the Chinese. And I wonder if you've got any thoughts on why that would be, or what might be driving that difference. Why would you expect one ethnicity to be quite distinct from the other two?
Right, Peter. I mean, if we go and we look into the ancestral origins, as well as, like how the populations moved or migrated and the original locations, it does seem like the Malay population that we have is very close to the Chinese population. and the variants are similar. In even if you take into account the Singaporean population, we know like the certain practices of the diet as well as in general, their phenotypes are way more matched than what we see with the Indians. The type of adiposity, the fat deposition that we've done through MRI studies, they are much more matched between the Chinese and the Malay than the Indians.
Okay. Okay, now I think there's some questions coming through here. Let me just get those up. And so the first question from Michelle Hill, do you plan to compare Asian genotypes and lipotypes to Caucasians and African?
Great question. We've already started doing it, and there's an ongoing work on lipidomics that we are doing with Peter's group, and we do find quite a bit of overlap, but we also see some distinct profiles in the two populations, which are also linked with the metabolic health of the individual, specifically BMI. That's what we've looked into. But we are having an ongoing work for comparing the genotypes, the variants that we have in our population with the Australian group too. So yes, there's ample, and then we've done work of comparing our variants with what's available from the previous Human Variome project.
Okay, that's good. The next question was,
Peter, can we take those other questions at the end? We're just running to time at the moment.
Okay, all right. So there's a couple of other questions there on the Q&A list there, Nirja, which you might answer in the next little while. You can type your answers in, I think, but we'll come back to those at the end.
We should have some time at the end for some open discussion, so
yep,
we can get to those first if that's cool.
Sure.
All right, I'll pass back to Anna then to introduce the lightning talks.
Okay, thank you, Nedra. That was really nice. So now Tusi is sharing the screen, and we start with 6 lightning talks. And our first speaker is Clara Castaños, who's going to talk about honeybee nutrition. Clara, the floor is yours.
Thank you. Hello, I'm Clarissa. I'm part of the Honeybee Health Research Group at UWA, and today I will talk to you about bees and their nutrition. So a good nutritional condition is indispensable for the health of the whole colony and for the productivity of the hive, which is translated into money for the honeybee industry. So as part of my PhD project, I'm investigating the nutritional decline of honeybees, and I'm correlating this, I'm investigating the lipidomic profile of honeybees, and I'm correlating it with hive measurements that beekeepers can detect in their hives. So for this, I set up an experiment where I provide differential feeding to 3 groups of hives. The first group I fed with sugar that contains mainly carbohydrates. Then a second group I fed with sugar and pollen that contains carbohydrates, protein, and lipid, what is a complete nutrition for honeybees. And I have a third group that I left into starvation. The experiment consists in 2 periods. First period where I did these differential diets for 4 weeks, followed for a recovery period from the week 4 to 25 weeks. And I analyzed the physiological condition of the bees with lipidomic analysis from the week 0 to the week 8, covering these diet treatments and some part of the recovery period. But I continue measuring the hive indicators until week 25, so I can track the performance of the hives. Here are us doing every week the hive measurements and taking samples of the bees and how we provide the differential food. This is a pollen paddock with sugar. Next slide, please. Okay, as hive indicators, we measure the quantity of brood, And the brood are the capped cells in the frames, which contains the baby bees that had not hatched yet. And as you can see during the diet period, the quantity of brood of the starving group that is represented in red declines, but then in the recovery period by week 6, it recovers again until 21 weeks. They fully recover that the other, like the other diet treatments. It was slightly a different story for the height of the weights. As we can see by the week 4 of starvation, the hives of the starving group decrease in their weight, but then in the recovery period, even when they recover, they never fully recover as the other 2 treatments until week 25. So this is demonstrating that starvation has a long-term consequence in the productivity of a hive. Using liquid chromatography in an orbitrap spectrometry, we identified about 200 lipids in the software MS dial, and from these lipids, 165 were significantly different between our groups. A linear discriminant analysis plot from these lipids shows that there are clear differences between the lipidomic profile of these 3 treatments. Then a heat map of these lipids comparing the starving and the sugar and pollen treatment showed an increase of a group of lipids, a decrease, sorry, of a group of lipids in the week 3 and 4 that are the weeks of really starvation. And this group of lipids were mainly triglycerides. Then another heat map of a FAME analysis of fatty acids shows that there is an increase of some fatty acids for the starving group at week 4 of starvation. A decrease of triglycerides, which are the main long-term energy storage lipids, shows that at starvation, bees are catabolizing these macromolecules into fatty acids. And in periods of high demand of energy, these fatty acids are being used as energy source, and also they are increasing in the fat cells as we are measuring. Now we are currently analyzing other proportions of the lipidomic profiles that we are developing into an assay for the early detection and measure of malnutrition in honeybees. And this will be useful to improve the management strategies for the beekeeping industry. That's it. Thank you very much.
Thank you.
Thank you. Just as a reminder, we take all questions for the lightning talks after the last lightning talk. And you're all encouraged to punch your questions and comments into the Q&A box. And you can do this either under your name or also anonymously if you feel shy, even though there's no need to feel shy about it. So our next speaker is Felipe Martelli, and he's going to talk about the effect of insecticides on Drosophila. Over to you, Felipe.
Thank you. Can I share my screen, or because I have quite a few animations to go through? It will be quick, but if I share my screen, it might be easier.
Sure, I'll share in. Yeah.
Thank you, Tulsi. So, hi everyone. Today I'll be talking about how low doses of a neonicotinoid insecticide can impact insect biology. Our data have recently been published at PNAS, and if you'd like to know the full story, here are the paper details. I'll start by giving a brief background on the problems that we discuss in this work. So, around the world, insect populations are crashing, with grave consequences to agriculture and ecosystems. This problem is caused by a combination of factors, and one of them is the intense insecticide usage. Insecticides are designed to be used at high doses in target pest species, but we have almost no idea of what they can cause at low contaminating doses in non-pest insects. So with that in mind, we try to understand the molecular mechanisms behind the impacts of low insecticide doses in insects. Then, using the model Drosophila, we observed that low dose of imidacloprid, which is the most used insecticide on the planet, cause a low but enduring calcium influx in nicotinic acetylcholine receptors in the insect central nervous system. Even though this low dose has no impact on survival, we believe that by increasing calcium influx, it can create mitochondrial damage and oxidative stress. In fact, after 1 hour of exposure, we observed a great increase of ROS in the brain, which created a cascade of damage. It leads to an increase of superoxide also in the anterior midgut, increase of lipid stores in the fat bodies, which are the insect liver, reduction of lipid stores in the Malpighian tubules, the insect kidneys, and also in the midguts. And we also observed changes in lipid levels in hemolymph, the insect blood, and a systemic reduction of ATP level, and also mitochondrial aconitase activity, which is a ROS-sensitive enzyme, which indicates, once again, oxidative stress in mitochondria. Along with those alterations, we also observed the severe change in gene expression in the brain and fat body, mostly in genes related to metabolism and also mitochondrial activity, and also a severe reduction in the expression of immune response genes. We also checked the lipidome of whole larvae exposed to these low insecticide doses, and we observed a reduction of phospholipids, mostly phosphatidylethanolamine and phosphatidylcholine, which are the most important phospholipids in Drosophila membranes. And they are also very important to regulate the lipid environment because they can affect lipid droplets dynamics. And we also observed a severe reduction of cardiolipins, which is a group of lipids we can only find in mitochondria. So once again, supporting the mitochondrial impairments. I then checked the effects of chronic exposures using a low insecticide dose that kills 50% of flies in 25 days. Here, when I treat these insecticide-exposed flies also with a powerful antioxidant, which is AD4, survival increased from 47 to 83% by day 25. One of the most important things that I checked in adult flies was the visual system. And flies, they have a compound eye, and each one of these clusters here is an individual eye or nomatidian. The photoreceptors at the center, they are surrounded by glial cells, which are vital for neuronal function. And these glial cells, they are dying in exposed flies. And if we check the mitochondria, we can also observe the mitochondria show a lot of impairments. So all this weird crystal here in mitochondrial membrane defects. So once again, supporting oxidative damage and mitochondrial impact. So in summary, we found, we described the mechanism that is centered in the production of ROS that triggers neurodegeneration, blindness, and also all those changes in the lipid environment. And nicotinic receptors, which are the targets of imidacloprid and also targets for most insecticides we use nowadays, are highly conserved in insects. And what is interesting is that most insecticides we know have some capacity to produce oxidative stress. And that's super interesting because this model that I described here is potentially conserved for other insects and also for other insecticides. And is the first model to explain how low doses could be contributing for this insect Armageddon that we are living. And finally, even if they don't kill the insects, this very low dose of insecticides can definitely play a role in this insect catastrophe because they can synergize with all the other impacts and all other threats that insects are facing, like climate change and parasites. And with that, I would like to say thank you, especially to collaborations at the Baylor College of Medicine, the University of Melbourne, where I did my PhD, and also Metabolomics Australia for all the help with the Lipidomics data. And thank you very much.
Thank you, Philippe. Our next speaker is Nicola Gray on the metabolic phenotype for COVID patients.
Thank you very much, Anne. I think Tussie's just loading the slides.
Nice music in the background. Is that yours?
There we go. Perfect. Thank you so much. So it's an absolute pleasure to be part of the virtual podium today. I'm talking to you from the Australian National Phenome Centre, which is located at Murdoch University in Perth. And for the past few months, we've really been dedicating a lot of our analytical resource to exploring the metabolic phenotype of SARS-CoV-2. One of the great things about being in Western Australia has been the very few positive cases that we've had, but that's also meant that in terms of research projects, that's been a bit of a challenge. So you'll see here the numbers of our positive cases are relatively low. We have got longitudinal samples for those, and we've matched them against people who were sick but negative for COVID-19, and then healthy controls as well. And our metabolic phenotyping has been on the plasma of those people and profiled using a combination of both NMR and mass spectrometry. So the results that you'll see on this slide are all from the mass spectrometry data that we've collected, and this has been carried out using a targeted approach for amino acid metabolites and related metabolites, particularly those involved with tryptophan and kynurenine metabolism. This was kind of highlighted from our early work as being particularly important in the disease, and it's also been highlighted by a number of other researchers around the world as well. And you'll see in the top center picture there, the OPL-SDA, which clearly shows the separation based on this chemical phenotype that we can clearly see between the healthy and the positive patients. The corresponding loadings plot just to the right of that illustrates which of those metabolites that we've measured really are driving that degree of variance. So you can see here, definitely those that are involved with the tryptophan metabolism pathway are either highly correlated with the healthy or the positive, showing that they're really responsible for driving this separation. So the aims of this study were really to try and understand a bit more about the mechanisms underlying the disease, really because it is such a recently emerging disease. There's still quite little that's known. So anything that we can find out may help with future patient treatments, and also in predicting who of those who have the disease may go on to develop worse or more severe cases so that they can be treated much better. The little box plots in the bottom left just really show the most significant metabolites that were perturbed between the healthy controls and the positive group. And again, it's these real sort of key metabolites that we're seeing involved with the tryptophan metabolism pathway, as well as some of those that are suggestive of perturbations and disturbances in liver metabolism, as well as kidney disease. So the little summary slide that you can see at the bottom right of the screen there really just highlights the fact that what we're seeing here is not just a respiratory illness. It's a metabolic illness that's showing signs of disrupting liver metabolism, kidney metabolism, and also having the potential to have a big impact neurologically as well. So in terms of the tryptophan metabolism, some of the metabolites that we're seeing has been significantly changed, toxic. So quinolinic acid and 3-hydroxykynurenine are neurotoxic compounds, and we're seeing those highly elevated in patients who have COVID-19. And that has obviously quite potentially detrimental longer-term health effects as well. Can I have the next slide, please, Tussie? Next slide. Thank you. So this publication here was released just earlier in the year, a few months ago, from our group, and it was a combination of this NMR profiling together with targeted mass spectrometry. And what was really significant here in terms of the NMR data was this severe change in the lipoproteins. So with COVID-19, patients were showing much lower HDL, upper lipoprotein A1, lower HDL triglycerides, and that was in combination with high LDL and VLDL triglycerides. So this is really pointing us towards perturbations in lipid metabolism. So in addition to that metabolomics that we've been carrying out, our next steps are really to look at the lipids in a much more sort of detailed picture. So we've been developing a method for the last few months to allow us to quantitatively target as many lipid species as possible. So we've been optimizing a methodology here based on the SciEx QTRAP 6500 in combination with a liquid chromatography separation. And that's allowing us to monitor several hundred lipid MRMs in both positive and negative mode. So this is the next step in terms of our COVID research, is to apply this to really highlight in more granular detail the lipid changes that we're seeing as possibly being associated with COVID-19. And I've just got a few acknowledgements there from my colleagues at the AMPC, and thank you all very much for organising the event.
Thank you, Nicola. Our next speaker is Shirley Guan, and she's going to share her screen. She's going to talk about the microbial lipidome. Shirley, over to you. We cannot hear you yet. Shirley, we cannot hear you.
Can you hear me now?
Yeah, now we hear you. Yeah.
Hi. Hi, everyone. So I'll be talking briefly about my lab's research on microbial lipids, and we are interested in the topic of microbial lipids because they make up, microbes are actually a big component of our body, and they are in homeostasis with us, but they also cause diseases. And as you can see from this cartoon, bacterial lipids are more complex than human lipids, as justified by the cell wall of gram-negative and gram-positive bacteria. And because of the foreignness of the lipids, they are actually recognized by toll-like receptors that can cause immune cell signalings, but also variation in lipid structures can be very potential drug targets. So there are a lot of fundamental questions that we still don't know about bacterial lipids, including things like how many lipids are there, what are they, how are they regulated, and what are their functions. And specifically, I'll show you one of our research on antimicrobial resistance. So what is also very disturbing to us is actually, if we plow through the literature, there's actually huge discrepancies in the lipids reported in microbes. Using the example of Acinetobacter baumannii, which is a gram-negative bacteria found in soy, but accounting for 5 to 10% of all hospital-acquired infection. So summarizing what we found in the literature across 4 papers, on the x-axis is the number of lipid species in the classical phospholipids with their abbreviations, and the number of species that represented in the different publications. So you can see that there's actually a great discrepancies in the numbers and the types that have been reported. And what is also concerning is that whether these identifications are actually precise, because if you talked about biomarker discovery or pathway discovery, this would be a major concern. And again, flagging one of this study, they actually found a very long phosphatidylserine in the bacteria, and usually bacteria don't make such long chains. Furthermore, they also found sphingolipids, and it is pretty well known that sphingolipids are unique to eukaryotes with few exceptions in bacteria. So this motivates us to do much better for in terms of microbial lipidomics development. And what our ambition is actually to establish a panel for microbial lipids to establish this as a reference library. And we have selected 6 species of bacteria, which are WHO priority pathogens, in collaboration with Sean Vasu from National Center of Infectious Diseases. So the 6 bacteria species that we are focusing on is Enterococcus faecium, Staphylococcus, Klebsiella, Acinetobacter, Pseudomonas, and Enterobacter. And they are called ESCAPE, not just because of their name, the abbreviations of their name, but actually they are known to escape the antimicrobial milieu, and they are critical threats because of the development of drug resistance. So we use mass spectrometry, of course, to probe the lipidome, and wanted to understand variations. So what we do in the lab is to first perform the kinetics of these different bacteria species across multiple batches to understand batch-to-batch variations and also growth variations. And this is a principal component analysis of the lipids that we identified in Acinetobacter baumannii with over 200 lipid species consolidated. So you can see different patches in the PCA analysis. And the good news is actually that we are able to perform highly robust analysis in bacterial. As you can see that this variation is mainly due to the natural variations of bacteria during their growth phases. Within the growth phases, you can see the different colors of shading, and these are actually only batch-to-batch preparation that is done across different weeks of experimentation. And we have also established long-term reference and QC controls for this reference panel of bacteria. So with this technology in place, we can potentially screen bacteria to understand their phenotypic differences in terms of antimicrobial resistance. And one application that we have is to study the roles of lipids in polymyxin resistance. So polymyxin resistance can be attributed to variations in the lipid cap of gram-negative bacteria, specifically variation in lipopolysaccharide or lipid A. And polymyxin actually is a cationic peptide that binds to the negative charge of LPS. So sadly for patients, a patient from the Singapore General Hospital, he was initially infected with a Acetobacter baumannii, which was polymyxin sensitive. And later on, the clinicians isolated the same species of bacteria which became resistant to polymyxin. Given the known fact that polymyxin interact with lipid A, which is on the cell surface, we actually then screen the lipid profiles using this robust method. And we can see that this big shift of the lipid profiles. So in summary, we have actually developed quantitative microbial lipids with high definition identification and link lipids to antimicrobial resistance. There are a lot of other applications that one potentially can go into. And with that, I thank you for attention.
Thank you, Shirley. Our next talk is Boeng Chung on the cold tolerance of wheat. Boeng, over to you.
Thank you, Anne. I'm still waiting for the slides to be changed. Thank you, Anne, again, and good afternoon to all. My name is Boeng. I'm from the University of Melbourne and currently working at the University of Malaysia, Sabah. Today, I would just like to share with you a bit of my PhD project entitled Phenotyping the Chilling and Freezing Responses of Young Microspore Stage Wheat using Targeted Lipidomic Profiling. We go to the introduction. Frost events have led to severe yield drop to the Australian wheat industry with around 360 million of losses annually. And therefore, this makes the improvement of chilling and frost tolerance an important trait for breeding of wheat. So my study aims first to use a metabolomics and lipidomics approach to understand the metabolic trait in a cold acclimation potential of two spring wheat varieties with a contrasting cold tolerance, followed by the identification of a potential metabolite and lipid markers for chilling tolerance in wheat. And finally, to determine which tissue, either the black leaves or the spikes, is more suitable for large-scale cold tolerance phenotyping in the future. Look at the figures on your right-hand side that shows the dilemma of that faced by the Australian wheat farmers almost every year since my PhD there. The yield harvest yield of wheat has been seriously affected by spring frost events that happened, particularly in the Western Australia from 2016 to 2020. So as a scientist, what we can do to help the farmers, especially in the aspects of breeding of more cold tolerant cultivars. So my approach, my methodology is, please look at part A, the experimental setup. We set up experiment first in the control environment using two wheat varieties with different cold tolerance, the young cultivar and the wild Kacham cultivar. Using the simulation, by simulate what the real frost events that happen in nature. The most important point here is the time point 1 referring to the short cold stress, which is equivalent to a short frost events, a sudden frost events, and the prolonged cold stress, time point 3 is equivalent to a continuous frost events. And the tissues types that we are using is the black leaves, which is the most important source tissue of the wheat, and also the whiskering tissues, the spikes, which is the most important sink tissues of the wheat at the young microspore stage or the reproductive stage, since this stage is very sensitive to cold stress. And we are using a targeted lipidomics approach, measuring the phospholipids and galactosyl lipids using the triple quad, LC triple quad approach. Next slide, please, to see. Yeah, so these are some of the main findings that we found quite interesting. The first one is the cold-tolerant variety young shows significant higher unsaturation to saturation ratio compared to cold-sensitive wild caution after prolonged cold stress in both of the tissue types, the flat lips at your left-hand side, the time point 3, and the spike at the middle for the time point 3. So we postulate the possible mechanisms that response to cold of the with tissue, the membrane is that after expose or during exposure to a harsh low temperature stress, the cell membrane will tend to increase the unsaturated species with double bonds that introduce kink structures to their structure to split apart the lipids right and left. This is important to for the cell membrane to maintain its fluidity in cold environment. So we also identify some potential lipids. That's around 13 lipid species, and they are all unsaturated species of them here. So the key message of this table of these 13 overlapping unsaturated species is that it's not only useful to distinguish between the two types of wheat varieties, but also distinguish the two types of tissues. So in summary, remodeling of membrane lipids via the control of unsaturated lipid levels are believed to be the key to maintain membrane fluidity during low temperature stress, which also distinguish between the young and wild caution varieties. And between the two tissue type spikes and flat lips, there are interesting distribution of unsaturated species between them, which complement each other and hence can be used interchangeably for cold tolerance phenotyping in large scale in the field in the future. Thank you.
Thank you, Wang. Our final speaker for today, lightning talks, is Young Hong on LVL in rheumatoid arthritis and CVD.
Okay.
Young Hong, over to you.
Can you hear me?
Yeah, we hear you very well.
Okay. Okay. And then I start. Okay. Good. Can I share my screen?
Yes, sure, sure.
Okay, and good afternoon, everyone. My name is Yong Hong, and I am now a second-year master student in Taiwan, Kaohsiung Medical University, and my major is medical laboratory science and biotechnology. And I'm very appreciated that I can have a learning talk here, and the title of my presentation is Investigate the Characteristic of Low-Density Lipoprotein in Rheumatoid Arthritis Patient for a pilot study of the cardiovascular disease. And first, the rheumatoid arthritis is a chronic inflammatory autoimmune disease that causes cartilage and bone damage due to high-degree joint inflammatory responses. And this disease is commonly found in women than men, and especially in the middle age of the patient. And from previous findings show that the rheumatoid arthritis usually accompanied by the extra-articular symptoms such as the cardiovascular disease and usually leading to the death of the rheumatoid arthritis patient. And L5 is one of the subfraction of the LDL, and L5 is the most electronegative low-density lipoprotein and is includes higher level of LPC and ceramide when compared to other subfraction of LDL. And alpha is a non-atherogenic factor that causes the cardiovascular disease, but its etiology for cardiovascular disease in rheumatoid arthritis patient remains unclear. So therefore, identifying the characteristic of low-density lipoprotein that contribute to the cardiovascular disease in rheumatoid arthritis patient may be crucial. So for our short result for the biochemical profile, we found that for we found that the CRPs is significantly increased in the rheumatoid arthritis patient, while the low density lipoproteins is decreased in the rheumatoid arthritis patient. And by using the fast protein liquid chromatography, which is the FBLC, to separate the LDL, and we found that for the control, the L5 is very low, but for the RA patient, the L5 is apparently elevated. And this is the quantitative result. We can find that for the control, the L5 is only 0.39%, but for the RA patient, the L5 is 1.81%. And this is the we convert this quantitative result into figure, and we can observe here that the L5 is significantly increased in the RA patient. And we further use the LCMS to observe the LPC in the patient, and we also found that LPC is increased in the rheumatoid arthritis patient, but for the ceramide and spingarnine is decreased in the RA patient. And so our short results show that the L5 and LPC may play a role in the pathogenesis of cardiovascular disease in the RA patient. And we will further do a further study to explore the mechanism and the role of the L5 and LPC in the rheumatoid arthritis patient that contribute to the cardiovascular disease. And that's all for my presentation. Thank you.
I didn't even get to ring my little bell.
Thank you, Young Hong, and also thank you all to all 6 lightning speakers. We now have some time for a joint Q&A. So if you perhaps wish to switch on your cameras again, and then we can discuss together. I see that there's a question to Nicola on 1 second. The question is from Julia Grasso. She's asking, a positive group, was there a difference between symptomatic and non-symptomatic, or did you maybe have too few cases?
Yeah, great question. For this particular cohort, we did. There wasn't a huge range in the symptoms that were presented. All of the participants that were tested positive did have symptoms, but we're subsequently expanding this to some more international cohorts. And in that, we do have a range of different severities and a whole array of symptoms. So once we've processed all of that metadata, we'll be able to probably tease out from like the really preliminary results that we've seen. We definitely see that some of the metabolites that were significantly increased in our cohort are definitely associated with severity. So in more severe cases, they're far more significantly increased than in those that have got sort of less severe symptoms.
Okay, thank you. Is a question from Margaret Coe to Boeing. How did you decide which lipids to target for the analysis? When you are on mute.
Sorry for this. Thank you, Anne, and thank you, Margaret, for the good question. So for the measurement of lipids, since we know that the plasma membrane is the first barrier for any biotic or abiotic stresses, for example, the low temperature stress. And that's why what is the most abundant lipid classes that mostly abundant in the plasma membrane will be the phospholipids. Okay, so that is our first target for lipids for measurement. Yeah.
Thank you.
Anna, I've got a question.
Go ahead.
Could I ask Philippe? I wanted to follow up a little bit on the ROS that you induce in response to the insecticide, and so where do you think that's coming? Is that coming from mitochondria activity, and how does that relate to the changes you saw in the triglyceride levels?
Yes, so most likely comes from the mitochondria, because what happened is if you have excess of intracellular calcium, you basically have two mechanisms happening. One is you activate calmodulin that activates nitric oxide synthase, produces nitric oxide, and then that's a ROS that will damage mitochondria and lead to more ROS. And then if there is too much calcium, mitochondria can basically uptake this excess calcium in the cytoplasm to prevent other damages. But what happens is the calcium inside mitochondria literally will poke holes in the inner mitochondrial membrane, which creates more instability and then more ROS production. So most likely calcium influx created by the insecticides binding to the channels and opening the channels for a very long time is what causes all this damage. And then we believe that at some point, this ROS contained in the brain will break free from the brain, basically in the form of peroxidized lipids. And there are some lipid transport particles in Drosophila that can carry those peroxidized lipids to the other tissues like the midgut or the mammalian tubes, etc. In terms of triacylglycerols, we didn't see great changes. We observed, of course, some changes in triacylglycerol, but because we did a whole lipidomics, we think that some of those phenotypes were sort of masked because we saw huge increase of lipid stores in fat body, but at the same time decrease in mammalian tubules and midguts. So if we had done, for example, a very target-specific lipidomic analysis for different tissues, we would see more of a difference there in terms of triacylglycerols.
Mm-hmm. Thanks.
Just got 1 minute left. 1 minute left, Anna.
Clara, can I ask you for the bees? Can you? I missed the part on how many bees do you have to pool to analyze the lipidome, or can you look at individual bees? And if it's individual, do they differ from each other?
Yeah, actually, each of my samples were 20 bees. So we went to the field to sample every week during 9 weeks. From each hive, each week, we sampled 20 bees. So we have like more variability in the hive because they are super big.
I see.
Yeah.
It will be interesting to see if individual bees differ from each other or if they're all the same.
Yeah, but this is more in catch experiments, but we want to see the response of the whole hive. That's why we also just sample baby bees. So we are sure we are not sample the gut and what they eat that day, just the recent hatched bees.
Thank you.
Mm-hmm. Thank you.
And that's our time as well for the lightning Q&A in this session.
Okay, our dear timekeeper, brutal. Now, questions and also, and if you all agree, we would now move on to the second keynote speaker and address all remaining questions later in the joint Q&A. So I hand over to Peter to introduce the second keynote. Thanks all.
Okay, thanks again, Anna. And yes, again, my pleasure to introduce our second keynote, Professor Masanori Arita. And so Professor Arita is from the National Institutes of Genetics and is also a team leader of the Metabolome Informatics Research Team at the RICAM Centre for Sustainable Resource Science. And so his team really develops software platforms that are then used for metabolomic analysis and network analysis and computer simulations, as well as designing databases for more efficient identification of metabolites. So Professor Arita is really a leader in this field, and I think the software that's been developed by him and his team has really been used extensively, both in Japan and in many other groups around the world, to integrate analysis of metabolomic and transcriptomic data. So it's my pleasure to pass over to Professor Arita for his talk.
Thank you for introduction. I'm Masanori Arita, and my background is computer science. So I'd like to introduce the computational aspects in metabolomics and lipidomics research. Well, on the first slide, I wrote some of my motivation to enter this field. As a computer scientist, I'm more interested in dissemination of information to the general public. I want to empower all researchers so that everyone can perform as a leading researcher in the world. Most important part for identifying more metabolites is data sharing in this sense. If everyone can start reusing very good data, then everyone has a chance to find good discovery. And what are the best way to enlarge or activate the entire community in that way? Well, provision of good data and software platforms together for free. So that's my research motivation. And I started the first metabolomics related project, Mass Bank, in year 2006. At that time, there was no free spectral database for MS. Nowadays, there are lots of databases, commercial and free, but at that time, none. So we started this Mass Bank project, and now Mass Bank servers are one in Europe, one in the United States, Mass Bank of North America, and the original one in Japan. And last year, I actually delegated the whole system to the Mass Spectral Society of Japan, MSSJ. So now the society is maintaining this Mass Bank server. And after this database, I started the MS Dial and MS Finder project under the bilateral collaboration with Professor Oliver Finn at UC Davis. And as introduced, the software MS Dial is now very widely used. And the main developer of this software is Hiroshi Tsugawa. I have been mentoring him from his student years, but now he's quite independent. He's writing all the software codes, and he will become a PI early next year. So he will become independent too. And since 2 years ago, I am responsible for the DDBJ repository. Actually, DDBJ is a DNA sequence repository, but I am putting one service to this DDBJ activity, which I will introduce now. Oh, my slides are not proceeding. Oh, I don't know why. But let me share the slides again. Now I'm sharing the screen again, and page doesn't proceed. I don't know why.
Yeah, you're right. We can
Maybe
just
try moving to the next slide before you share. It might have.
Yeah, I'll do that. Well, my local machine, the slide proceeds. So this is the second slide. I would like to compare genome mining and metabolome mining. Maybe genome mining is a much older word. Actually, the word genome mining was used in the free issue of Nature published in year 2000, reporting the Human Genome Project. In that Human Genome Project issue, I remember there were close to 10 research papers mentioning genome mining or gene mining in the human genome. And I'd like to introduce, likewise, metabolome mining to the community. Basically, we can retrieve raw data from a repository like metabolites or metabolomics workbench in the United States. And then we should be able to predict molecular formulas and molecular structures and use that information for hypothesis building to obtain biological discoveries. However, we stumble because in genome databases, all sequences are freely available, and you can run BLAST and other software programs to analyze such sequences. But in metabolomics, there are 6 major vendors using 6 different binary formats. And when you come to the analysis part, you stumble again because there are so many software programs, but you need to learn Unix and you need to learn how scripting languages and so on. So that's the original motivation why we started creating MS Dial, MS Finder, those all in one platform for metabolomics. The crucial part of metabolite identification is the structure prediction. And for structure prediction, the most important part is the prediction of molecular formula. Well, it's common sense, but it's really important. That's why I'm re-emphasizing this issue. From accurate molecular formula, you can narrow down candidate structures to, say, 10 or 20 from the PubChem database, from the PubChem structure database. So accurate prediction of molecular formula is the prerequisite. And for this purpose, we can exploit various technologies or techniques. For example, in order to estimate the number of carbons or number of nitrogens, quite often we use isotope labels. You can dope isotopes, say carbon-13, and analyze the whole tissue or whole sample using the same mass spec platform and compare the original or undoped sample to identify the number of carbons or nitrogens. And for the number of hydroxyl groups, you can utilize the derivatization in GCMS technology. I will explain this technique in the next slide. And from the MS information, you can identify neutral losses, and those neutral losses correspond to molecular fragments like typically hexoses or acyl derivatives or others. So using these key techniques, now you can estimate molecular structures from mass spectra. And this is what MS Finder does in our software all-in-one platform. And MS Finder is actually rule-based. When you hear about rule-based technology, it's a very, very old technique. When you ask a computer scientist about rule-based system, they will say, oh, it's old tech of 1970s. Nowadays, many machine learning is available, but we actually stick to this very old technology because we want to know the rational steps of fragmentation. And we want to use that understanding for a better structure prediction. That's why MS Finder is still rule-based system. And the rule MS Finder is using is basically the even electron rule. Even electron rule is a very old idea about stable ions. Usually, stable ions possess even electrons, not odd electrons, because radicals will react very fast. So in order for mass spectrometer to observe, stable ions must exist for a certain time length, time window. So they should be even electron ions. And MS Finder basically uses this rule, and we extended this rule to sulfur and phosphorus atomic elements, in addition to the original nitrogen, oxygen, and carbon system. That's why MS Finder works, or MS Finder outputs comparable results to machine learning based software programs like CSI FingerID or others. And why our rule-based system can work so good, as good as, say, machine learning based or AI based software programs? That's because our data are still very noisy, and also because our knowledge is far behind that of genomics, so to say. In genomics, there are so many cleaner data, and so many people have been working on that data using AI and support vector machines. That's why those data are now very well curated and applicable for those high-tech computer programs. But in metabolomics, our raw data are still noisier, very much, yes, not in a large scale and with a lot of noise. So MS Finder still works. So our aim is to curate metabolome and lipidomics data so that AI or SVM programs can perform better than our rule-based MS Finder. This is an example of metabolome mining. This is a bit old paper, almost 3 years ago, but with a curated metabolome repository called Beambase at Oliver Finn Laboratory, UC Davis. We first searched for unfrequently appearing but non-identified metabolites. And we selected several of them, one from cancer tissue and others from feces, basically from gut bacteria. And for those selected samples, we isolated or we fractionated the corresponding part and reanalyzed those unknown metabolites using GCMS and LC-MS-MS. And the reason why we use GCMS is through derivatization, we can identify the number of hydroxyl groups in the unknown structure. And from LC-MS-MS, by using MS-Dial and MS-Finder, we narrow down candidate structures from the database. But here, we not only used PubChem database, but also we used the MINE, hypothetical structure database. MINE database is with another repository Professor Finn has been creating. And in this database, around 400,000 structures are generated, hypothetically generated using a very common enzymatic reactions from well-known primary metabolites. And by querying candidate structures in this hypothetical structure database, we could select the correct structure, in this case, N-methyl UMP, as a cancer-specific metabolite, not known to be in PubChem repository. So with a good database and with appropriate software programs, we could prove we can identify a new molecular structure, not only not from already known structures, but from a hypothetical unknown structure. So in order to extend this approach to a larger scale, now we are creating a metabolomics data repository called Metabobank. Now I delegated Mass Bank to the Mass Spectral Society of Japan. So now at DDBJ, we are opening a public repository for metabolome raw data, just as Metabolites is doing in Europe. And our Metabobank repository just opened last month, and now we can accept your raw data, but the input interface is written only in Japanese right now. The user manual is in Japanese, so I will create the English version very soon. But what I can say is our repository is permanently maintained with a permanent identifier, just like metabolites in EBI. So now this repository completes my kind of not complete, but complements my goal. Now we have a raw data repository and a software platform, and you are ready to analyze your data together with already published information. So this is the last slide. I want to emphasize the three important points in the structure prediction or structure selection. One is molecular formula is very, very important. Without accurate molecular formula, you cannot predict. You cannot write a good paper. And second, please use more dimensions, not only retention time and mass spec, but nowadays collision cross-section is coming. And I hope Professor Zhenjian Zhu next week or maybe 2 weeks later, he will mention this CCS information. His laboratory is very strong, and we have just published a joint paper in Nature Biotechnology this year. And the last one is please use experimental methods to know more about chemical groups, not only neutral losses, but as I said, if you derivatize using TMS, you can estimate the number of hydroxyl groups. Maybe by using other agents, you can estimate the number of, say, amine groups or other chemical groups. This is very important for structure prediction. Thank you for listening.
Thanks very much, Masanori. That was really great, very interesting. And we now open this up for questions. And just while we're waiting, perhaps I could start. Your mining example that you showed, it looked like you pulled together a lot of samples to do that mining. So it was over 100,000 samples that you pulled together, which is a massive feat. You must be congratulated for that in itself, just to align those. But my question is really is, you know, do you think this is the way to overcome some of the limitations that you mentioned earlier by creating bigger data sets and by doing, you know, meta-analysis of multiple data sets? And the follow-up question to that then is your Metabobank, will that automatically integrate data sets and combine data sets?
Well, I think more data provide more accurate information, because in standard metabolomics, we still see much noise. So although we think metabolite peaks, maybe not most, but many of them are noises. It's possible. In GCMS, it's more reproducible. So usually we run many samples and align those samples to obtain a cleaner GCMS spectra. But in LC, we cannot do that much. So a better way is to integrate more data. In this particular slide, Oliver Finn has been accumulating huge samples in his laboratory using almost the same measurement method. And Beambase is the original name for his spectral repository. And this Beambase, yes, contains thousands of tissue samples and also microbiota samples. And we first selected quite frequently appearing spectra from among those, because without such frequency or abundance, it would be difficult to identify structures or biological effects or I would say, I don't know, biological effect or biological meaning of those metabolites. That's why.
Mm-hmm. Yeah, okay. There's a question on the Q&A session, which was, firstly, great talk. Can MS Dial use the NIST library for identification?
Yes, NIST library is, of course, yes. Yes, okay, yes. And also from our RECAN website, team website, we make many spectral information for free. Not NIST, but our own measurement data are all available. And curated spectra from Mass Bank and other repositories like GNPS are also public from our RECAN data resource. So please Google search using the keyword MS Dial. Then you will hit our prime website, prime repository from weekend. And my plan is to integrate, sort out this information and integrate all spectral information into our Metabobank soon.
Yeah, and so just coming back to the Metabobank, again, so is that a platform to bring data sets together, or is it a platform to put your own data set in and then mine that data set? So again, it comes to this thing of alignment of data sets. And I understand now in Oliver's case, of course, he'd run all the samples, so it was probably relatively straightforward to do the alignment. But when you get data sets in from different people, it might be another matter.
Well, personally, I'd like to standardize data in this Metabobank, but it's a public repository. So basically, please accept all data submitted. But from among those submitted data, I plan to curate some large data sets so that mining will be possible.
Yeah. Yeah, no, and look, certainly in the lipidomics field, that's, I think we're looking towards how we can bring big data sets together. But again, it's challenging. That's going to be one of the big challenges, I think.
I agree, yes.
Okay. Any other questions from the panel or the audience?
We could also start opening the floor to questions to all speakers now. Yes, for the last few minutes, and there's been from Nedja's talk. There's been 2 questions still which haven't been addressed yet. Nedja, great that you're still there. Thank you very much. So there's a question to you saying, how do you differentiate population stratification from QTL mapping when comparing people of different ethnicities? Did you answer that yet?
No, I did not. But great question. I think population stratification is just not QTL. There is an agreement there. There's only a part of it that can be explained by QTL. So there are other factors related to health and risks, disease risk, which can be diet, lifestyle, socioeconomic status. There are many elements to it, which we have been capturing and incorporating in our analysis of predictive modeling for diseases along with the QTLs.
Actually, Nedja, could I ask you another question as well on your the arachidonic acid and the FADS gene and the different proportion of the C and the G, I think it was. So when you showed that the genotype, the proportion of variance in the different ethnicities seemed to align really very exactly with the proportion of arachidonic acid in those ethnicities, and which was nice to see. But I would have expected there would be multiple variants in FADS that would be contributing to this. And so did you find a lot of variance in FADS? And so, but you seem to have, but that particular variant was just the strongest or the lead, or what was the story there?
Yes, Peter, it's a 200 megabases region, and it has way more variants. It's just not one variant, but this one variant is the tag SNP for the haplotype that we have there, and is a strong determinant. So if we use this, it accounts for a majority of the SNPs that we see in the FADS locus.
Okay. Yeah, that makes sense. Okay. There was another question, I think. Thanks for a great talk. I think this was also for Nedja. I noticed that the PCA plots had different PCs being plotted rather than the first couple that explained the most variation. And I think, yeah, you had a plot where you had PC 7 and 8, or was there a reason for this? And could the data type used, e.g. 0, 1, 2 for genetics methylation continuous between 0 and 1 and transcriptomic continuous, also have affected the clustering?
Yep. Very important question indeed. Sorry, I missed on those details because of the time limits. But if you look into genetics, there's very less technical confounding. And when we separate and we look into the top PCs, it is the ethnicity pops up as the topmost. And so you would see that's the PC2 that came up in the plot that I showed. Now, when you go into epigenetics, it's the cell type composition. There's a lot of variants that has been seen, and they come up as the top PCs there. So the top 4 PCs there are from the technical variation that is in bisulfite conversion, and also what we saw for the cell type composition. And once we correct for that, and now we take into account all the phenotypic measures that we had, including ethnicity. So we took these women's BMI, their mental health, their other statuses. Among them, ethnicity still ranked to be the topmost and explained the most variance that we were seeing. And so if you see that the PCs are not necessarily the same as the genetics analysis, that's because of the technical variation that we encounter. And that's very well known in the field that those are the top PCs that dictate the data.
Mm-hmm. Thanks, Nidra. I don't see any other questions there. Any other questions from the panel? Have we still got Nicola on the line?
She had to take off. She's going to answer via email if she can, I think.
Okay.
Yep.
We are a little bit over time already, so we could be mindful of time and finish by thanking again all speakers and all participants for this really thoughtful presentations. And Tusi, shall I share the screen and show next week's?
Yes, Sani. Yeah, go ahead. Yeah. Thanks.
So today was the second virtual podium, and next week we have a third. Here you see the agenda, and again it's 2 keynotes and 6 lightning talks, again from a wide variety of different topics covering COVID, crop sciences, and various pathophysiologies. So I invite all of you again to sign up for this and join us next Friday. So with that, a round of applause to all speakers and panelists and participants, and thank you all very much.
Thanks, Masanori. Thanks, Nidra, and all the lightning talks.
Thanks, everyone. See you next week.
Thank you.
Bye-bye.
Bye.
Bye.